Okay, good afternoon, everyone. So I'll talk about my lab. We started like a year ago, and our primary interest is how nucleus contributes to different epithelial behavior. If you think of nucleus and genetic information, imagine this piece of paper as holding all your genetic information. If you try to fold it into a piece of ball like this, you can't read any words because there are no words on this sheet, but the kind of words that you will read from this piece of folded paper versus if I remodel it and change it again, and the kind of words you will read it in the next folded form are different. And if you imagine DNA packed into nucleus, in similar analogous form, the kind of genes you will read from a particular form of nucleus will vary from the form of the nucleus in different contexts. And because you are packing this nucleus into this form, it also acquires a material property at this stage that is not that you cannot study at, let's say, polymer scale of DNA. So we are interested in this material behavior at this scale, how it contributes to epithelial behavior at a cellular scale, and how it contributes to how genetic information are being read, and how it contributes to the fate of the cell depending on this form it acquires. So generally, how do you see the behavior? How do you infer physiological relevance of material property versus genetic content? In for material property, if you take a epithelial cell and detach it from a surface and allow it to readhere back to a surface, the height that a cell can flatten up to is limited by how much you can flatten your nucleus, and you can push it further, and the nucleus is you can sorry about the lag. So you can push the nucleus further, but it does not really go. You can artificially push it, but it maintains a certain tissue height, certain nucleus height and cell height. And when you move from an isolated cell to a collective environment like epithelial cells, in flatter epithelial cells, the area of the cell is limited by how big your nucleus can be. And in more relatively thicker tissues like pseudostratified epithelium here, it affects how much cell you can pack in a unit volume. And because epithelium in a developing context is subjected to so many different mechanical behaviors of folding, it's easy to imagine when when you have to go through any folding event, you have to move the bulky nucleus out of the way. It's not in, yeah. So whenever any epithelial event happens, you have to move that nucleus out of the way. And if you time resolve this process, if you time resolve this process before apical constriction begins, actually you see nuclear displacement towards the towards the basal side in this particular case. And if you play around with this material property of nucleus, don't let either you. Sorry. If you play around with this material property, either don't let it be further squished. Give me a sec. It's lagging behind a lot. Okay, this is the slide we are focused in. And I'll wait for the video to play. So if you perturb with the material property of the nucleus, in this particular example, you're not the nucleus is basically rigid and it stays in a three-dimensional uniform sphere, and it is not compressible like control environment. And we are looking at a Drosophila gastrulation event, the process is affected. So if you play around with the material property, how cells intercolor during gastrulation gets changed. So they have mutated a protein called KUK's Kugelkern, and it's a lamin C type protein, but it's not lamin C. It's one of the lamin related proteins. So the nucleus just remains rigid and round. So if you play around with the nuclear property, it does affect tissue dynamics, and it can put a lot of epithelial morphogenetic events associated with these dynamics. And if you zoom out from this material property and zoom into the genetic information encoded in the chromosome of in the if you zoom into genetic information stored in the nucleus, based on how you're deforming your nucleus, because chromosomes occupy different territories in the nucleus, you stretch different parts of DNA. So different parts of DNA get expressed, and different kinds of gene get expressed based on how you compress or stretch the nucleus. And towards this line, if you take a nucleus and allow some external load on it, there is a global change in genetic expression or repression. And some of these kind of experiments has led to this idea that a different mechanical environment or different form of nucleus connects to different kind of genetic architect or chromatin organization within the nucleus. And if you take this one step further, if you not just compressive environment experience within the epithelium at different parts, if you force the nucleus through a narrow channel, in principle, you can control what kind of fate emerges from the cells. So this is a really beautiful experiment. You can take fibroblast cells, which are differentiated cells, not of epithelial origin. You can expose them to certain growth factors here through some lentiviral exposure. And when you induce expression of those genes, you can convert them into induced neuronal cells, and that happens at this efficiency, it's less than 5%. But if during this period of exposure to growth factors versus actual induction of those growth factors, if you expose them to a narrow compressive environment by passing them through channels like this, either 5 micron or 7 micron diameter channels, the efficiency of this neuronal conversion increases a lot. So a lot of these evidence kind of point towards the mechanical changes in the nuclear form, shape, can affect cell fate acquisition. And that's what we are primarily interested in doing in the lab. And we are trying to understand how, when DNA is packed into this compact form from a thread-like organization, how the material property changes based on the size and shape of the nucleus, and how does it affect the local cell behavior. And keeping these two aspects in mind, we are interested in looking at two different kinds of epithelial tissue. First, how it leads to emergence of pseudostratified architecture in epithelium, and what happens when you push the nucleus to a really extreme cell scaling boundary, and how does that contribute to epithelial tissue development. So for the first part, we are looking at pseudostratified epithelium, which is different from, let's say, some of the simple epithelium we have been looking at. Simple epithelium is when you have your nucleus all at the bottom anchored to the basal side, and you have a nice epicobasal gradient in this. Stratified epithelium is when you have multiple layers of cell. A nucleus can be basal or at the center of the cell. Pseudostratified epithelium is unique in that way that it's just one layer of cell, but the nucleus occupies multiple different positions within the cell. So this is, I'll touch upon the diversity of the tissue, physiologically where you find this tissue. It's found in the developing tissues of many of these organs across these species. And Drosophila also, I think I'm not showing here. It's also found in proliferating tissues in adult tissues like reproductive organs in mammals, and some other proliferative, unnatural or pathogenic proliferative conditions like salivary gland papillae and tubular adenoma, which are cancerous version of epithelium. The epithelium has undergone cancerous transition, but it does not lose its epicobasal polarity. The cells just become pseudostratified. Here, the nucleus is not visible, but these light blue structure a nucleus in a healthy epithelium. And this is how it looks like in a cancerous epithelium, which has undergone this pseudostratification transition. So this kind of epithelium is found where you have where you have high proliferative properties, and you have a need to store a lot of these cells. So, and this is how they look when a pseudostratified epithelium is undergoing division. Yeah. So here we're looking at a mouse cochlear epithelium. The black objects are all the nuclei, and you can see some of the nuclei going to the apical side and dividing and getting reintegrated back. And sometimes the nucleus marked in magenta or green, they kind of they show stalling behavior. They do not move completely to the basal side before the next round of cell division. Yeah. So cells are constantly dividing. They're moving to the apical side, but not always you find cells coming back to the basal side. Sometimes they're just getting anchored or stalled at different positions. And when you increase number of nuclear layers in a pseudostratified epithelium, you're basically adding more and more cells, and you're increasing the storage capacity of the cell. And there is some correlation between the position of the nuclei and the cell division, cell cycle stage. So if you think from a mitosis point of view, you will find mitotic cells at the apical side, G2 cells moving towards the apical side, G1 and S cells coming towards the basal side. And for a long time, people have thought that this architecture, this pseudostratification, is a passive outcome of asynchronous cell division across different cell types. But there are enough experiments where you can stop cell cycles and subsequent process, and but pseudostratification doesn't go away. And you can stop this process of nuclear movement to apicobasally during interkinetic nuclear migration. That all part of cell cycle, but it does not get rid of the pseudostratified architecture. And you do find this architecture in non-proliferative tissues like retina. So we have been thinking of what does this architecture do, and us and along with a couple of other labs, we have been thinking of this architecture as a storage architecture. This pseudostratification, moving the nucleus onto different vertical planes, is allowing you to store more cells in a unit volume. And in principle, it increases the proliferativeness of the tissue because you're sharing the same apical area between more cells. So we are trying, and if you think of this pseudostratified architecture from a nucleus material point of view, you can also explain local folding events like here in intestinal morphogenesis, where wherever the new villi is going to emerge, the nuclei are pushed to one side, to the basal side here. Or you can also explain global tissue curvature events where you can push all the nucleus to the basal side and give rise to curved tissue like optic cup. So we are trying to establish that pseudostratification is a storage architecture using mouse and embryonic stem cell organoids. And in this context, we're trying to perturb with nuclear shape and form in many different ways and see how it affects the packing fraction or storage capacity of the tissue. But this is something, an artificial system we're trying to establish. This is an architecture with organoid. We're curious about if you sample through different pseudostratified tissue across many different organisms, can you see some of these behaviors naturally happening? And when we went through many different organisms, Drosophila imaginal discs are a really useful tool here. And we found out Drosophila eye, like other imaginal discs, is pseudostratified. Briefly, this is the Drosophila epithelium, eye epithelium. This part gives rise to antenna, this part gives rise to adult eye. And this epithelium goes through a process of morphogenesis, where morphogenesis starts from this posterior and it travels in a wave-like manner to the anterior side. You have this apical, this wave is made up of apical constriction of undifferentiated cells. So the differentiation wave basically passes from the posterior to anterior side. And this wave appears as an apical constriction in the epithelium. Before the constriction, you have all the undifferentiated cells. After the constriction, you have differentiated cells coming out. And in that differentiated cells, you will have a segregation of photoreceptor versus non-photoreceptor. And if you look at the cross-section of this epithelium, yeah. So this is how it looks from the top of from the top view. This is the differentiated part of the epithelium. The differentiation wave goes like this. You see apical constriction in a wave-like manner. These are undifferentiated cells. And it is pseudostratified along this whole epithelial tissue. And I don't want to play any more videos because it's slowing down shit. But okay, so this constriction basically passes in a really beautiful way. That's important to see. If you look at pseudostratification in this tissue, there is a marked difference between the epithelium on the either side of the morphogenetic furrow. Till it comes up. So this is the eye epithelium. We're looking at this part of the epithelium in a cross-section view. This is the furrow. Here you can see the enrichment of actin and an apical devoid of the apical part devoid of any nuclei. This is pre-MF, this is post-MF. Here you can see the number of nuclear layers or pseudostratification complexity, if you might say, is different across the morphogenetic furrow. You have less number of nuclear layers pre-furrow. You have more number of nuclear layers post-furrow. And if you quantify the size, different, let's say, form-related properties of the nucleus, the area of the nucleus is higher in the cells pre-furrow, and it goes down when the cells enter the morphogenetic furrow, and it remains smaller in the differentiated part of the epithelium. And similarly, the shape of the nucleus, it's more elongated in pre-furrow cells, and it's more roundish in the furrow versus in the furrow as well as the differentiated part of the epithelium. So the question is, how are you reducing the cell or nuclear area suddenly? If you look at time-lapse movies of this developing epithelium, as the cells enter the furrow, they go through a cell division event and you see a sudden change in nuclear areas. Hopefully the video will play soon. So here we're looking at, again, the same view of the epithelium. Furrow is progressing like this, and these cells are entering the furrow. And if you zoom into this furrow part, you see cell division events happening right at the boundary of this morphogenetic furrow and let's say, undifferentiated eye epithelium. And we have been trying to quantify what, if you track these morphological changes of the nucleus during this progression of the furrow, can you correlate some behavior of the nucleus? Can you correlate some of the morphological features of the nucleus to the differentiated state of the cells that come out past the furrow? So can we find out mechanical features or morphological features of nucleus pre-furrow? And does it correlate with the acquisition of fate here, photoreceptor versus non-photoreceptor differentiation? Here, the furrow acting as a mechanical compressive environment. And does that compressive environment, is it getting experienced differently by nucleus across the furrow? And does that contribute to acquisition of fate of photoreceptor, non-photoreceptor past the furrow? So that's what we're trying to do with the live cell imaging. And if in this, and if you look at this, the image on the left again,
I recall correctly, the fact of being a photoreceptor is a property of the pathogenic gene.
Yeah. So the molecular aspect of this epithelium, how fate comes up, is known really well. So we are trying to characterize if those behaviors correlate with some of the let's say, morphological features of the nucleus, and can we predict those behaviors before even cells enter the furrow or while they're going through the furrow?
I think that there is more from the lab of Sarah Bray, where she looked at the shape of the nucleus versus the expression of notch, for example, notch reporter gene. I was wondering whether this would make sense with your model. I think she has been looking at the deformation of the nucleus and the expression
of the
notch target genes.
Yeah. So I'm not familiar with that work, but I'll read about that. But I feel like it would contribute how the cells are reading the morphogenetic cues in the furrow. It would contribute. I imagine it's a nucleus which is, even though the morphogenetic furrow here, all of them are G1 arrested and all of them should be at the basal position, but they are not linearly at the basal position. So the exposure of nucleus to concentration or amount of the morphogen would be different based on where the cell is, where the nuclei is present. I have a feeling maybe from there we can connect how much morphogen cue you read, and depending on that, what is the fate you acquire. So that's what we also want to do. Another interesting thing that
You talked, well,
part of the
do you get
So I don't have those mutant lines yet, but we are trying to get those lines and see how the whole eye development is going to look like. I imagine it won't go through normally because the folding events in that mutant would be very, very different from normal epithelium where you can compress the nucleus. I don't know if in that work they comment on survivability of the organism to later parts. That was an embryonic stage. So if they survive, the eye epithelium development would be really nice to look into. Yeah. Past the morphogenetic furrow, when the cells are getting differentiated into photoreceptor versus non-photoreceptor, within this pseudostratified epithelium, which is one layer, we start seeing segregation of nuclear position towards the apical or basal side. We start seeing this tiny gap here, this kind of gap, and it is consistent across the differentiated part of the epithelium, and that gap corresponds to segregation of photoreceptor versus non-photoreceptor. You can stain that. You can confirm that with ELAB staining, and because we are interested in this mechanical aspect of nucleus in morphogenesis process, we looked at is the segregation happening from a mechanical point of view. And if you look for lamin C expression in these post morphogenetic furrow part where cells have gone differentiation, photoreceptor cells have higher lamin C. They are more rounded. They are more rigid in they are more rigid based on lamin C staining. Lamin B seems to be normal, but lamin C only corresponds to those apically segregated nuclei. And this kind of lamin C expression appears only after the furrow has passed. It appears in the differentiated cells, I assume. Yeah, so the lamin C expression and segregation of this nuclear layer starts post the furrow. Yeah, so here we're looking at lamin C, and it only, yeah, it appears after the furrow. Lamin B does not look any different across the segregated nuclei. And if lamin C generally works, this mechanical aspect of lamin C works through link complex, and we're curious if other components of the link, other members of the link complex are also involved. If you look at CLAR, which is on the outer nuclear membrane and binds to nuclear lamina through another intermediate protein, that seems to be a opposite localization. It's not enriched in photoreceptors which have higher lamin C. It's enriched in non-photoreceptors which have less lamin C. So here, I think CLAR isoforms could be playing different kinds of role, and we're going to look at that. Eventually, we had just started with this project. So the idea is to get a nuclear metric, which involves many of these physical or morphological parameters of nucleus, characterize this whole thing across the developing eye epithelium, and see can we predict the acquisition of photoreceptor versus non-photoreceptor fate during this morphological changes as the nuclei experiences through the furrow. So now we have a interesting line, dendra 2 line, so we can photo convert into green to red and track individual nuclei. You can photo convert one nuclei and track through the furrow and see where it ends up and characterize their mechanical and morphological environment of the nucleus as it goes through this chain. So this is what we are trying to do with the Drosophila eye morphogenesis. But if you compare pseudostratified epithelium across many different species, just as a factor of nuclear morphological features and mechanical properties, can we find some general rules that explain the tissue-specific behavior? So we have not done anything yet, but a glance at the published literature, one thing that appears is whenever neurogenic fate is emerging or developing in different organisms, you increasingly experience a let's say more compressive environment across different species, be it mouse or zebrafish or Drosophila. So the chances of or the occurrence of neurogenic fate is always correlated from past studies. Is correlated with higher nuclear packing, higher complexity of pseudostratification. So we are curious about, does the nuclear compression play any active role in this neurogenic fate? And we're trying to dissect these role of contribution of growth factors versus morphogen and in nuclear compression. So that we are trying to address with organoids again. You can generate retinal organoids from mouse embryonic stem cells. And based on how you, based on which time during the, let's say, growth of the organ, at what point you expose neurogenic factors, we can play around with that timing, as well as we can get rid of the compression by stretching, keeping the organ under constant stretched environment so that you don't acquire that compressive environment, even though you have the morphogenic factors. So it'll be nice to see if lack of compression leads to less neurogenic fate cells appearing in these tissues. And that's what we are trying to do. Yes.
Well, simply, I think it's really cool that you have the compression as it goes, and then that might be related to gene expression. Thinking about the fact that the nucleus might be in any given position, right? Relative to the chromatin and specific genes that might get exposed or crashed. Are you thinking of ways to?
Yeah, so in the Drosophila eye development, so when the nucleus moves up and down, we see in this actin spot constantly, let's say, anchoring the nucleus to cytoplasm or the lateral cell membrane, and that actin spot does make an indentation into the nucleus. I think as it's moving up and down, it interacts with different kind of chromatin and probably locally alters which chromatin is there, and maybe that helps in photoreceptor.
Do you think the position of the chromatin is stereotyped somehow?
I think there is some external role in anchoring specific type of chromatin to specific part, and based on as it goes through the furrow, if it is in an open state, it will probably, hypothetically, it may give rise to a photoreceptor. But if it is not, then we are trying to characterize in that spot what are the chromatin and what are the genes that are anchored at that point, and if that varies between photoreceptor versus non-photoreceptor. Yes.
To make the connection between nuclear shape and transcription, don't you need to have a readout of transcription?
Yeah. So we have not started that yet. So after this characterization of nuclear form, we are going to characterize what are the genes, like not all of the genes, but the genes that are readily known for photoreceptor versus non-photoreceptor, and see transcription-wise which kind of, along the pseudostratified layer, where do they correlate? Yeah. Yes, that's the next step. That's what we're trying to do with pseudostratification. It will be nice to perturb this architecture actively, where we can move the nucleus up and down and perturb the nucleus and local compressive environment. And we are in the process, we are going to test these constructs to either actively displace the nucleus or anchor the nucleus at specific parts and see how it perturbs local mechanics and tissue behavior. This is something we are, there are some genetic ways to achieve this, but we'll first try with the mechanical ways and then come to these genetic perturbations. Other than the role of nucleus in pseudostratified architecture and how it contributes to different morphogenetic events, we are also interested in looking at nucleus when it reaches an extreme scaling region. So Malpighian tubules and salivary glands of lepidopteran insects, they are, unlike other morphogenetic events where you divide and give rise to more number to grow, they grow in cell size with multiple rounds of endoduplication. The cell number doesn't change, only the cell size keeps increasing. And this growth happens during larval stages. This is how the Malpighian tubule in Flutella looks like, and the nucleus looks like at early larval stages. And when it enters into L2 and L3 or later larval stages, the cell size increases and the nuclear volume increases, your nuclear content increases, but the shape of the nucleus undergoes drastic change. Instead of a round 3D sphere, it becomes extensively branched. And I don't have experiments here, but if you somehow damage the cells and the cytoplasm is open or leaky, the shape of this nucleus does not become round. It does not fall back to a round architecture again. It maintains this branched nuclear architecture. So we are curious if this branched nuclear architecture at this scale, does it contribute to the cell size or cell shape directly as a cytoskeletal element rather than just a genetic element? So if you zoom into it, specific parts of any specific part of this larger L3 nucleus. This is how the nuclei look like. And we are primarily interested in what are the new features, nuclear behaviors that emerge as a factor of scale. One obvious thing that you can imagine, how does transcription and transport of RNA look like out of these big giant nucleus? And if you stain for active transcription site, instead of small phase condensate vesicles that you see in typical nucleus, they form these extensive networks with bigger nuclear-like structure and thinner branches in between. So we feel it's like a way to achieve transport faster. We have to do more experiments towards this direction, but this is where we want to go. And these are the things we are interested in generally. How does nuclear material property contribute to tissue mechanics? And how does that also bridge the gap between tissue mechanics and the, let's say, cell-fate acquisition? With that, I'll end my presentation here. Let me just thank a couple of people. The Drosophila eye work I showed was primarily from Lulu and Ethan. And a lot of people have been giving me their insects, flies or moths or other things. I have been borrowing organisms. And everyone from Roland and some collaborators and some mentors and some cats.